Hello, everybody. Welcome to another Expert Talk series. Remember, this is teamwork. It's each of us. We have our speakers, but we also have each of you. And each of you know a whole lot about your personal life and the problems and the good things that have happened. So I'm glad you're here tonight that you can share with us after our presentation. And why don't we just get on with it?
Welcome, everybody, and thank you to everybody who could join this next installment of our Everything ALS Expert Talk series. Tonight's program is an update from the leadership of Prilenia, presenting on Pridopodine for treatment of ALS. We're incredibly proud to have them here. We've been working with them for some time, and they've been super dedicated not only to the advancement of central nervous disease science in ALS, but as well as Huntington disease. And without further ado, I'm going to introduce them a little bit and give some background on Michael and Henk. So, Michael Hayden is the Chief Executive Officer at Prilenia, and prior to this role, he was President of Global R&D and Chief Scientific Officer at Teva from 2012 to 2017. During this time, he led the approval of 35 new products in major markets, with many for diseases in central nervous system disorders. Michael is a Killam Professor at the University of British Columbia and a Senior Scientist at the Center for Molecular Medicine and Therapeutics. And he's been recognized in Ken McGugan's book, 50 Canadians Who Changed the World, listed as one of the 50 Canadians born in the 20th century who have changed the world. Michael's a co-founder of 5 biotechnology companies, including Prilenia, Neurovir Therapeutics, Xenon Pharmaceuticals, Espriva Pharmaceuticals, and 89 Bio. And he currently sits on different public and private boards of biotech companies. Michael's a prolific author in science. He has over 900 peer-reviewed publications and invited submissions, and Michael has focused his research primarily on translational medicine, including the genetics of diabetes, lipoprotein disorders, Huntington disease, predictive personalized medicine, and drug development. Michael and his research group have identified 10 disease-causing genes, which include the identification of the major gene underlying high-density lipoprotein, HDL, in humans. And Michael also identified the first mutations underlying lipoprotein lipase, LPL deficiency, and developed gene therapy approaches to treat this condition, resulting in the first approved gene therapy product, Glybera, in a major market. Michael's the most cited author in the world on ABCA1 and Huntington's disease. An inductee into the Canadian Medical Hall of Fame in 2017, Michael has received numerous recognitions and awards, including an honorary doctorate of science at the University of Göttingen in 2014, the Luminary Award by the Personalized Medicine World Conference in 2014, the Diamond Jubilee Medal on behalf of Her Royal Highness Queen Elizabeth II in recognition of his contributions and achievements. He has many, many more awards, so I'll just call out a couple in particular that I thought were really interesting in recognition of his outstanding career achievements, including the Canada Gardner Whiteman Award, recognizing him as a physician scientist who's demonstrated outstanding leadership in medicine and medical science. And I'd also like to introduce, and Michael is committed, by the way, to really empowering others. One of the things that we've learned about him is that he's an amazing mentor. He's mentored more than 100 graduate students and postdoctorates, and he's also a TED mentor. So we're really lucky to have you in our space, Michael, and thank you for the many years of service you've provided. And joining him today is Henk Schuring, who's Chief Regulatory and Commercialization Officer at Prilenia. And he also has over 30 years experience in pharmaceutical industry. And Henk is a pharmacist with a proven track record in rare diseases and as a creative strategic thinker. He has worked on several first treatments for rare diseases and has been working in industry at various positions in regulatory affairs and in commercial at both regional and global levels. In his role at Prilenia, he plays a critical role in defining the regulatory pathway for marketing authorization and development and clinical programs. Michael and Henk both are wonderful thought partners to us as we think about innovative ways to make sure that we can help accelerate treatments out of just clinical trials and into the market. So we really welcome them both here. And at this point, I'm happy to turn it over to Michael and Henk to begin your presentation. So thank you.
Well, thank you, Christian. What my mother's not alive, but she would have been proud of that that introduction. So thank you. I'm so pleased to be a partner with this community. I've been very moved by it. I feel deeply grateful the stories that this community has shared with me has not only challenged me, my brain, but also grabbed my heart. And this is really an integration of heart and mind in trying to bring some hope to people with ALS in the hopes that people can have improved quality of life. I know so many of you are watching your own agony as you watch your own illness transform who you are. They may change you physically, but your soul and your spirit remains alive. And we at Prilenia are deeply grateful to be part of this community. And we are inspired by your courage, by your thoughtfulness, and actually learn from you all the time. So we're grateful for you as our teachers too, as we continue to bring our own imagination and creativity to try and find some way to alleviate some of the really difficult issues that you're facing every day. So I'm going to just start and share with you a little bit about where we are in Prilenia. And now I'm just trying to see where that slide is, and it seems to have gone. So I'm going to go back and just pull that up. Excuse me 1 second. Maybe Henk, you could say a few words while I find this.
I will do that. So first of all, I'm incredibly pleased to be talking with Everything ALS and the patients. In the end, one of the most important things that we can achieve with developing new drugs is having a drug that ultimately becomes accessible for patients they understand and can use and want to use. Because in the end, if it is good for the patient, it is good for everybody.
So I'm going to tell you a little bit about this drug, Pridopodine, why we have faith in it, and where we're hoping to bring this to people with ALS as soon as possible. Now, one of the issues, I'm trained mostly initially in genetics, and one of the issues that's very interesting is that the target for this drug is the sigma-1 gene. And we already know in humans that people who have a DNA change in sigma-1 develop ALS. And this DNA change causes a loss of function. And when there's a loss of function that leads to no sigma-1, you get a more severe ALS as a child. And if it's a partial loss of function, you get an adult onset. So this relationship between the type of the mutation and the impact on the protein is really important validation. We've got lots of validation. Here, for example, showing you how this drug is actually interacting with its target. Sigma-1 is in the brain and very much in the motor neuron of the spinal cord. And this is using a label that labels sigma-1. And what you can see, it goes to the door and knocks on the door of sigma-1. And here you can see the whole of sigma-1 lights up throughout the brain. And one way to know what your target is to treat with your drug and see how it blocks this. So here we treat with Pridopodine first, and then you give the drug, and no longer can this label get to the door because it's blocked by Pridopodine, which proves that this is really the target for this drug. The sigma-1, when it actually is active, is neuroprotective, particularly in motor neurons, but throughout the brain, and has impact on a whole set of pathways that I won't go through in detail. But for example, one of the organelles in the brain that's very important is the mitochondria. It's the breathing part of the cell, and this is improved in function, and numerous aspects of function are improved. The end result is that neurons are protected. And for example, when we look at animal models of ALS, what we can see is that Pridopodine prevents muscle fiber wasting, prevents body weight loss, improves motor function, improves all kinds of functions that are important, secretion of a neurotropic factor that is protective. An improvement also is in motor neuron survival, which is really what we want. So these animal models were very helpful in confirming this, and this is done in numerous studies. Now, I just show you this because one of the key proteins that is elevated when the cell is stressed in the motor neurons is a protein called BIP. And all I'm showing you here is that when you use pridopodine at different doses, and particularly here at an important dose, it reduces it. And furthermore, when you look at the actual, these are other markers of progression, but look here, this is promoting cell viability. So it brings cells back to life in a sense. If you pre-treat with pridopodine, and here, for example, would be a cell that has stress, and now you can see at this particular dose, you see 71% of these cells are viable. So it promotes viability of the neurons, and that's really what we're looking to do. Importantly for all of you is this drug is safe. It has a side effect profile that's similar to placebo, and that's very, very important. So let me share with you, it was because of this that the people at the Healy at Mass General, and we're really thrilled to collaborate with them, chose to include this in one of the first trials as part of the platform trial. And just to show you, in the platform trial, people got the active dose and placebo was shared, and this allowed more rapid recruitment, and it was enhanced efficiency. Now, just when you look at this, what we saw in our particular patients who had definite and probable and also had onset less than 18 months and had fast progressors. And here you can see placebo shows that people are doing worse, but we saw very nice improvement even occurring at 8 weeks. So this was already very good. And here, if we look at the group that we are going to go into our trial with, and I'll ask Henk at the end to comment on the regulatory path. This is looking at people who had definite and probable ALS and less than 18 months duration. And you can see even at 8 weeks, we see a separation and some slowing of progression. And we were very excited to see that. And furthermore, when you look and see how this is contributed, here's the improvement. On the right-hand side, here is the improvement. On the left-hand side would be no improvement. And you can see the measure for measuring progression in ALS, the ALS FRSR improved, and it's contributed by all of the respiratory, bulbar, and motor function. And at every time point, 8, 16, and 24 weeks, all of these contribute to the improvement. What you can see here, for example, this is showing 95% confidence limits, and this shows you that the mean difference in 95% people is always in favor of the drug itself. Now, respiratory function, and the reason is this particular gene is highly expressed in certain regions of the brain, particularly the brainstem, and the brainstem is important for respiratory function. And here we see quite remarkably in placebo, of course, function goes down over 6 months, but here in respiratory function, you see maintenance of function with less respiratory deterioration, even over 6 months. In that measure, a key measure is shortness of breath, called dyspnea. And what you can see quite remarkably in this particular group of definite, probable, less than 18 months duration, we almost see no deterioration in respiratory, in shortness of breath. And this, again, shows tremendous slowing of decline in respiratory function. It's not only respiratory function that is showing this, in particular speech. And this has big impact because the brainstem is also important in speech, and speech depends on function of the diaphragm, the pharynx, the larynx, the throat, and the tongue. And it's measured by looking at different measures. This is measuring how fast you can articulate and also the rate of speaking. And here you can see there's already an improvement, and I'll just show you this. And of course, speech is associated with overall ALS progression, and we can know how important it is to maintain contact, to maintain community. And so this was something we were also very pleased about. So when we look at the articulation rate, after 16 weeks, it can go down quite profoundly. But here we can see in this particular treated group, they really remain similar to baseline with almost no progression. And this was also seen in speaking rate associated with this. And we're working with Everything ALS also to define what meaning this has and what clinical meaningfulness would be associated with some of these changes. Overall, what's most important is the quality of life is better than seen without the drug. So here you can see overall as measured by the ALS Q40, a well-defined measure for ALS quality of life. And we see it's much slower deterioration in total eating and drinking, and of course, communication. All of this would be dependent on respiratory function, pharynx, larynx, and tongue. With this, we were also interested to know whether this had impact on survival. And here you can see in the first 6 months of the study, 24 weeks or 180 days, there was no real difference. After 24 weeks, however, everybody went on to Pridopodine. There was a group that had been on Pridopodine from the beginning. We call that early. And these are people who were on placebo and went on to Pridopodine. And what you can see, if you were treat longer, the blue line is below the red line. And this means that survival, there's less survival if you treat it later with Pridopodine than early. One way of testing this is to ask, when do 50% of patients no longer survive with this? And what you can see, it's around 300 days. But at this stage, if you were on it already for nearly all the time for those 300 days, you only 15, so 85% of people are really surviving. And when you keep on going and look at when it's 50%, it's another 10 months, 600 days. So there's almost a doubling of survival in this subgroup. And that's also very important. So we have shown that the survival analysis showed encouraging positive trends. We saw some benefits in speech. When we looked at everybody in the population, every up to 3 years duration, it wasn't benefit because the time was too short. This included also slow people who are slowing. But when we looked at the group that was definite, probable, and early, there was significant benefits for progression, respiratory, swallowing, quality of life, and speech. And so as part of this, the Healy Center was looking to get immediate access to people for this drug because of the promising results. And this was a grant that was sent to the NIH. And I'm really thrilled to say this was funded. And now Pridopodine is already available in the United States and the first patients have been enrolled. This just came out at the end of April from the Healy Center at Mass General and Prilenia. And this is for people who've had 24 months of more duration of illness, because all the others will be in the phase 3 study that we're about to start. So I'm going to ask Henk briefly tell you a little bit about the path we have with the regulators, what they've told us, and then I think we'd be very interested to hear your comments and have questions from you. So Henk, maybe to explain the regulatory path right now.
Thank you, Michael. So let me give a little bit of the regulatory discussions and maybe also address some of the questions up front that I saw coming in. We went to the FDA, and let's talk first about FDA, with the data as Michael just showed. We had a meeting with them to discuss the data as well as to discuss a future phase 3 trial that we are planning. I've been in quite a few of these meetings, but I've rarely had such a constructive and positive meeting with FDA as we had on the data here. They gave us a lot of good feedback with regards to a future study design on endpoints, duration. But above all, what I really appreciated is we asked them a question if they would be willing to consider using speech as an important clinical endpoint in the study. And FDA clearly understood the clinical relevance for families and patients of speech and was very much willing to work with us on defining an endpoint for speech that we could assess in the clinical trial. We have agreed with them that we will go back to them to have some further discussions on this. And this is where everything ALS and MGH are very helpful in working with us on getting the further data to demonstrate this. But so far, it looks that we are able to get speech as an important endpoint in future clinical trials with Pridopodine. I do want to add that Pridopodine is orally administered twice a day, a capsule that you can swallow. And it is not intended for specific mutations. So it is not intended for patients with S1R mutations. It's intended for really a large group of patients that have ALS. And we are targeting for the clinical trial the early rapid. We have had similar meetings in Europe with the equivalent of the FDA, the so-called Committee of Human Medicinal Products, CHMP. And equally, they have been very supportive with regards to designing a future phase 3 study. And also they were very keen on including speech as a potential clinical endpoint. So very supportive, and we are on target to start working on getting the trial ready by the end of this year and maybe early next year, first patients in.
So let me try and, I see there are quite a few questions in the chat, and I just want to try and answer some of them, and then we can have more interaction. The first question from
Michael, we actually do a moderated session after, so
yeah.
So we will ask all the questions. So,
Please do. Okay, I can switch this off. Thank you.
You're welcome. Go ahead, Christian and Martha, who is going to be our moderators.
You're muted. Sorry.
Yeah.
I'm sorry.
Henk, was there anything else you wanted to speak to before we get into Q&A?
No, feel free.
Okay, great. I think one thing that might be just off the top might be helpful to explain that to further understand the promise of earlier detection and faster subtyping and how that could impact your ongoing, the future phases of work and what you learned from the trial so far. As far as the delay, the delay in onset or the delay in efficacy that you saw, to explain kind of how the endpoints interacted with the timing of the length of the trial. And you and I've had some conversations about that I think are really useful to explain to people what it means to make or miss a primary endpoint, but then see in post-analysis that sometimes you do see the signal that you're looking for, but just it's a matter of timing as well.
Yes. So just to say, our next trial will be in patients who have less than 18 months duration. But this does not mean that the drug may only be useful for patients with that duration. The issue with the people who have longer duration is they may be progressing very slowly. And so you don't have a window to see the effect of your drug. And we will learn from the expanded access program now, because these are people who are 24 months and more duration, and they're going to be followed for up to at least 2 years, but maybe 3 years. We will learn to see whether the drug is useful in everybody, irrespective of the duration. But because we want to get the drug quickly to patients, if this is going to be successful, we want to really look at those who are more fast progressors so we can see a signal earlier, so we can then get this drug to patients as quickly as possible. So this is also for everybody in ALS. This is not the protective regimens and the protective paths that are associated with it would be useful in ALS, but they're also useful in other neurodegenerative conditions, like, for example, Huntington disease, where we're already going for approval in Europe and in discussions with the FDA. We also have preclinical data in vitro and in vivo for other diseases like Parkinson's disease. But our initial focus is ALS and Huntington disease, and also a disorder called vanishing white matter disease, where we also have seen some early, very promising findings.
Great.
Thank you, Dr. Hayden. So we have another question, as you were seeing in the chat. So someone is asking about the sigma-1 receptor and how it's related, if it's related to the FUS gene. So could you explain a little bit of the difference?
Yeah. So the sigma-1 receptor is not related to FUS. It's present in these cells, and this is really like a shield. When you activate sigma-1 and it's activated, it's a shield for the brain and the spinal cord against many noxious stimuli. FUS is a very particular mutation where people are, Ionis and others are using antisense to try and knock that down and have impact on it. This would be quite different. This is a drug that's taken orally and would be appropriate for everybody with ALS because the mechanisms involved are generalizable to neuronal protection and not particular to one form of noxious stimuli.
Beautiful. Thank you. That's very exciting.
Thank you. And I'll take the next question. Is Pridopodine effective in C9 or 72 mutations?
Yes, we're still busy sequencing and we're waiting the data, but we would expect this to be significant in patients with all kinds of causes leading to ALS, because we're not dealing with the cause here, we're dealing with the effect. And the effect, for example, is decreased mitochondrial function, increased stress in cells, what's called ER stress. And this would be caused by many different types of mutations, and all of them would be having, hopefully, some improvement in the presence of pridopodine. So pridopodine is a shield below the cause of the disease. It's a neuroprotective mechanism.
Thank you.
Thank you. And to that effect with Pridopodine, Dr. Hayden or Schuring, what are the side effects that you're noticing, if any?
You want to take that, Henk?
Let me start with that. It's a very benign safety profile that we have seen. We are now busy in further establishing based on the various clinical trials, and things that we see are things like nausea, insomnia as adverse events that we talk about.
Yeah, but just to say that the nausea and the changes are no increase above placebo.
Correct.
So it has none of these side effects are greater than if patients were taking just a placebo drug. So it has a complete safety profile that's similar, that basically there's not a single either a mild event or moderate or severe adverse event that can be attributable to the drug. So we're very happy. We have 1,500 patients who have been on the drug for what we call patient years, more than a year. And again, the side effect profile is similar to placebo.
Wonderful. And I have a little bit of a question to that wasn't in the chat, but has there been much discussion about maybe different titrations of the dose, increasing the dose?
Generally, we start with a dose that's once a day and then go to twice a day over a week. So there is a very minimal titration over a short period of time. But again, even to go to higher levels quicker we have seen no serious adverse events, but we do recommend going slowly in the first week or so, and then the drug would be taken once in the morning and once in the evening for twice a day dosage.
Let me add something to that, Michael, because I think there was also potentially the question about higher doses with more efficacy, which is not the case. This drug has a very specific profile, and you really need to be at the 45 mg twice a day to maximize the efficacy. If you go to higher doses, we have shown that you will get less efficacy.
Okay.
And we know that, and one of the reasons is it's highly selective for sigma-1. which is probably what explains its safety profile. As you go to higher doses, you may get off-target activity on other targets that can induce less efficacy and also more side effects. So the dosage to be used is 45 milligram BID. We also know if you take one tablet a day, in other examples, you have a little less efficacy. So you want to get to a stable dose of one tablet twice a day. It's a small tablet and can be swallowed quite easily.
Great. Thank you.
Thank you.
Next question. Can we hope that Pridopodine will be made available in Canada through a program similar to the EAP in the US, the Expanded Access Program, for those who wish to access it?
Henk?
And that will have, we can always look for that. If we have questions, we have a policy on our website for expanded access. So if there are questions from Canada for expanded access, feel free to talk to your physician and have them reach out to Prilenia, as we are willing to support that. It will have to be supported also by the Canadian authorities.
And for our phase 3, our phase 2 study happened only in the United States. Our phase 3 study will involve Canada. And so, for example, we now already have expanded access for Huntington disease in the United States and Canada. And so we will be submitting an IND to Canadian authorities for our drug, and that would be one of the steps that could lead to expanded access for patients who are not eligible for our trial.
Thank you.
Thank you. And does the medication or Pridopodine, does it protect all the motor neurons?
Well, we believe that it's effective for all motor neurons, but it's going to have best effect in those areas where the sigma-1 is most highly expressed. That's the door it knocks on. We know it's very much in the motor neurons, but it's particularly the brainstem, also the cortex, by the way, and some aspects of the basal ganglia. But overall, it will have impact on all motor neurons, but particularly related to the impact on motor neurons in the brainstem neurons. And that also explains why we see most effects on respiration, speech, swallowing, and talking, bulbar function.
Thank you. And will this, as we talked about phase 3 a little bit and its expansion, will it continue to be on the Healy platform?
Of course, Healy will be a center in our study, but it will be global. It will be run through a CRO. This is now going to be a drug trial for approval. And so this will be also including Canada, the United States, and numerous parts of Europe. We're still working through exactly which countries, what we have to do. And we just had a clinical advisory board meeting this week where we had tremendous input from colleagues all over the world in the United States, Canada, and Europe. And we're finalizing our protocol, which we will also have a meeting with the FDA on speech, because they're very interested to really understand how we're measuring speech, how meaningful changes in speech will be. And we're hoping that this can be an important endpoint in our trial, because we know we have big impact on speech based on the prior data.
Thank you. So we have a question. We have a lot of patients and people that attend with ALS that are long-term survivors. We have someone who's been diagnosed for 20 years or 10 years ago. And what about people who have had long-term diagnoses of ALS? Are they eligible for the trial?
Well, the trial will be for people for, in all likelihood, less than 24 months or maybe even less than 18 months. So these people would be eligible for expanded access. And there are up to 40 sites, but certainly through Mass General, they're coordinating it to find out about a site close to you, because we are going to be looking at 200 people who are going to have access to the drug, who have had the disease for more than 2 years. And so This will be important for those individuals to be in touch, find out which sites are available. It's all throughout the United States, and these will be sites that they could enroll and get the drug now. And this, of course, will be without no cost and supported by the grant that's been awarded to Mass General and in combination with us.
I think it's important, Michael, to add here that it is very well understood that there are always clinical trials and eligibility criteria, and that some patients may not meet those criteria. And that's why this expanded access program is there, so that patients who do not qualify for the trial but still would like to get access can talk to their site. And as Michael said, there are about 30 to 40 sites that will be participating in this expanded access, so that patients can also get access to compounds like Pridopodine, even if they don't qualify for the clinical trial. That would be the phase 3 trial that we plan to use to seek approval.
Excellent.
We're very grateful that the US government and the NIH have chosen, after careful review, to support this for expanded access. And I think it was supported because the drug is safe. There are already some potential benefits demonstrated. And even though we will be doing a phase 3, we know that the eligibility criteria will exclude many patients with a longer duration illness, but now they'll be eligible for the drug. This will also be important for the community to see whether the drug also has influence and benefit for people with longer duration illness. And of course, just remember, the first study was only 24 weeks. These are going to be patients who are going to be followed for more than 100 weeks. So we'll be able to really learn and see what the long-term effects are and whether the benefits are sustained over a longer period.
You were reading my mind, Dr. Hayden. That's exactly what I was wondering as well. Thank you.
Thank you so much. I'll jump in with the next one. Is neurofilament being used as a biomarker, and are you recruiting any per manifest gene carriers?
So no, we're not at this stage doing pre-manifest gene carriers, but that would be certainly, once we've shown this is useful in people with early disease, it would be very interesting and important to see if we can delay onset in people who are pre-manifest. We are measuring neurofilament. Neurofilament did go down in the definite, probable, and early patients, and it will be measured as a biomarker in our study as part of this, but it would be an important secondary endpoint. Henk, do you want to add?
No, I think you said it totally right. It's an important endpoint, and we see more and more that regulatory authorities like FDA are keen to understand neurofilament in these neurodegenerative diseases. A trend in NFL in the right direction is exactly what they would love to see.
Excellent. Thank you.
And as we've said, yes, thank you. We've got a lot of sophisticated people as well on the call. We have one question. We've got someone asking, could you please let us know the survival benefits of the G93A ALS mouse model? I know it reduces ALS-induced cachexia, but no effects on onset and survival. Any comments?
Yeah, in those studies in mice, we did not see changes, any significant changes in survival. It's hard to really know. The mouse model itself is very severe, and we saw numerous changes that would predict improvement in survival, but we did not see that in the mouse model in the SOD1 mice that we looked at.
Very good. Thank you.
I'll take the next. And does this treatment only benefit ALS patients with known SR1 genetic mutations or other genetically caused ALS? You may want to clarify the relationship.
We showed before that if you lose S1R, you get ALS. That was in some of the human genetic studies. Here, of course, we're showing that if you increase sigma-1 using this drug, you see protection. This would be useful in all patients, not just patients with ALS mutations, but rather in all patients. The number of people with mutations in sigma-1 causing ALS is extremely small, tiny numbers, but it alerted us to a gene that's important for motor neuron survival and motor neuron function. And remember, those mutations all were loss of function mutations. Now we know if we have a gain of function through using this sigma-1 agonist or the sigma-1 activator, it acts as a shield against damage for motor neurons, which would be theoretically, and now we've shown already in the phase 2 study as part of Healy, that this may be beneficial for all patients. And of course, we're now doing the phase 3 study to confirm our initial phase 2 studies.
Thank you.
Excellent. Thank you. So we have another question. It says, can you elaborate a little bit and let us know in the clinical trial that we're talking about tonight, how many patients were on Radicava and Riluzole while on in the placebo group and receiving the Pridopodine?
Yeah, so we did not exclude people on Radicava or Riluzole, and we've looked at on or off, and there was no real difference. So this can be used with these other drugs. It doesn't minimize the effect of our drug, so it can be used in combination. Just remember, Riluzole and edaravone did not affect speech or respiration in the way that we did. So, but we also didn't see any particular added benefit, although there may be some that we still are busy studying to see whether the combination, there were small numbers in combination to really know. In our study that's upcoming, edaravone or Radicava and Riluzole will be fully allowed. We will probably randomize them so the equal numbers in both groups in an effort to make sure that there's not imbalance. And we will in larger numbers now, because remember the first study had small numbers. We're going to have in the close to 400 to 500 patients in the next study, and we'll be able to see where there's any added benefit of these drugs together, and we will be looking at that as part of the study.
Excellent. Thank you.
So we have a member of the audience who was in the Healy trial that just recently ended, and they're waiting for the expanded access to restart so they can continue taking Pridopodine for its benefits. Is there an indication of when that will occur?
Please, Henk.
The NIH grant has been given, and the expanded access program has started in the first sites. So this may be an issue at the specific site. And if the person would love to reach out, we can maybe contact and see how we can handle that. But the first sites of the grant expanded access have been set up and are allowing entering patients in the program.
And also, this is all the patients who were on the open label are being turned over to the expanded access program. And so we would expect as these sites open, but I think it'd be helpful to get back to us so we can facilitate this and support that transition where there may be some holdups.
Yeah. And thank you, Christian, for putting in the website.
Absolutely.
Thank you. And this somewhat dovetails on what we're talking about right now. We have a person here watching tonight says that they were on Pridopodine, the Healy D, for about a year and a half, and it ended 3 months ago, as we were just speaking about. It sounds like their symptoms stabilized. They were staying the same while they were on it, and since they've been off of it, their symptoms are returning. Should I go back on it?
Yes, I would hope that this person is fully able to go into the expanded access program at the site they're on. And certainly if they've seen advantages before, and we do know that it slows progression, has impact on other obvious signs and symptoms, it sounds like it would be good. And with a drug that has no obvious serious adverse event profile, it would be advantageous to go back and stay on the drug for as long as possible. Yes.
Thank you.
So we'll talk a little bit about the expanded access program in general. Is there any cost to being involved in the expanded access program?
No. The expanded access is fully supported by the government of the United States, and we are providing drug. And so this is being administered through Mass General and then through the sites. So there's no cost. I can't remember if there's any reimbursement for travel and accommodation if people have to travel, but all of these can be discussed with the site. But certainly the cost for seeing the physician and the drug itself are given and supported by the US government.
Right. As a follow-up question, somebody was offered the EAP. Would patients beyond 2 to 5 years of disease likely benefit from that?
Well, again, this will help us to learn more and help, but there's reason to be hopeful, and that's why the drug grant was funded. There's no reason that this would only be useful in the first 18 months of illness. If you slow progression, you would expect that to be slowed throughout. But this expanded access, firstly, because there's no real risk for taking it, we will be able to determine for sure whether there's benefit associated with slowing of progression against what one would normally expect for people on the natural history of ALS.
Thank you.
Excellent. Thank you. We have a question about Pridopodine. Do you anticipate possibly getting this fast-tracked designation from the FDA?
It's a great question, and we are going to look into that. If we are able to get speech as a clinical endpoint, and I saw a question in the chat box whether it was an exploratory or primary, we are looking to include it as a secondary endpoint, which would be key, and that would also give us a nice way to seek fast-track designation.
Excellent.
Also to say that we have got fast-track designation for Huntington disease in the United States.
Yeah.
So we have that. It's already been granted for one indication. But once we complete and get able to get everything, we will be applying for fast track for ALS. And we're hopeful based on the promising findings so far. But we don't have that yet.
Very good. Thank you.
Thank you. Is there any synergism of pridopidine with albuterol and clenbuterol that might make attribution of benefit due to pridopidine in clinical trials?
So we haven't tested that, so we don't know. The mechanisms, again, are totally different. So they, potentially could be, but I think those drugs also need further investigation about their benefit. We don't see any harm. So there's no harm associated with other drugs, but at the moment we have no clear evidence of benefit with any other drug at this time.
Thank you.
Thank you. You all have been presenting very nicely tonight about this, and as our audience, a lot of us realize the etiology or the origins of what causes motor neuron disease or ALS is multifactorial and highly complex, and they're looking for your comments. You're targeting the protein, the sigma-1 protein. Have you thought about targeting multiple proteins?
Well, the sigma-1 protein actually does target multiple proteins. So the sigma-1 is usually in an inactive state. When it is activated, it acts what we call like a molecular chaperone. In other words, it goes to multiple different sites and activates numerous proteins and has many pathways that are activated. So it acts on a single pathway. It's like a hub that then has downstream effects in numerous pathways. Remember, it has impact on mitochondrial function. It improves the mitochondrial contact with the endoplasmic reticulum, enhancing the mitochondrial respiration. It increases autophagy. Autophagy is a word that says removes toxic proteins. In ALS, they are aggregates that are formed in the cell this pridopodine, and we've got data to show that it reduces aggregates in those cells that can be toxic. It also enhances a protein that is very nurturing for neurons called BDNF. So it already acts on numerous pathways, and many of those pathways are disturbed in many forms of ALS, so that there may be different causes, but the end effect, as a neuron is moving towards a less viability, all these pathways are activated, and pridopodine has effect on many of them.
That's excellent. Thank you so much. That's and very exciting too that you're targeting it this way. Thank you.
And I'll take the next question. Does pridopodine have any effect on bulbar saliva secretions in ALS patients?
Yes, we are seeing some improvement in salivary secretions and in bulbar function. When we look at bulbar, one of the questions is salivation, and this is also improved. It was not significantly improved, so we need more. The numbers were small in that study, but the trends were all there for all aspects of bulbar function, including salivation. Yes, others have shown with sigma-1 agonists, with other drugs, that this might be also useful in terms of salivation. Yes. We will be looking at that, all these endpoints very carefully as part of the study.
Thank you.
Good, thank you. We have another question in the chat. It's what will be different in the phase 3 trial versus the phase 2 trial aside from adding speech endpoints? Can you speak a little bit more on that?
Well, I'll just make this and then let Henk speak. The first thing is the numbers will be much greater. So the first study, there was a total of 160 patients on placebo, 120 on drug, but the placebo was shared across different aspects. So there was only 40 people directly in the placebo for our trial. They were shared. We're going to have much greater numbers, between 450 and 500 patients in total in the trial. Secondly, the duration of the trial will be much longer. That first trial was 24 weeks. We are going probably double the time, 48 to 52 weeks. And then it'll be a blinded open label where everybody goes on the drug. But we'll have a longer time to look at progression, look at other features, and the numbers will give us greater power. Also, speech will really be hopefully an important secondary, and there'll be other features associated with what we've seen with sigma-1 function that'll be important associated with this. This will be done also not just in the United States, but will be done in Canada, as well as numerous places in Europe, which will also give us more generalizability in different geographies. At the same time, it poses more risk because different cultures have different ways of assessing patients, and we'll have to make sure all the raters are very carefully trained and in similar ways. Henk, you may have more to add here.
Well, not so much. You did a lot of the legwork already, but what I saw one question in the chat about the duration of the study and the duration of the placebo controlled phase, and I think it's important to clarify that. The study will be global, as Michael said, US, Canada, Europe, multiple sites, which is different, which means more patients. We are looking for a 48 or 52-week placebo duration. And then after that, all patients would be eligible to get on the drug for another year, which will allow us to look at, for example, survival after 2 years of duration of the study, and not too early, as that is an important element. Speech will be added as a secondary endpoint. But the key will be the number of patients will be larger, because we need to confirm what we have seen in the phase 2 study in a larger number of patients to seek approval at the regulatory authorities to ultimately make it available. Primary endpoint is most likely going to be the ALS-FRSR, and then the other endpoints will be the normal suspects that you have seen in many other trials, plus speech, as we discussed.
I'm going to jump in. I know we still have a number of questions, but there's still a number of questions about expanded access in general and how people can find if they have a site local to them that they could actually go through. The generic response is, of course, go to the website, the web page, and have the physician reach out. Is there any kind of list of Healy centers, part of the Healy network, or any others that people might be able to use as a resource as well?
I know that we are working on finalizing to get the this expanded access on clinicaltrials.gov, and that will include the list of sites. It's going to be there in the nearby future. Meanwhile, feel free to reach out to us, and we can connect you with Healy, who can help with that.
Excellent. As you know, we've got Nova in Florida and a number of other sites. So however best we can collaborate to make sure people know, we'll certainly help communicate when these updates.
But I do want to manage expectations, Christian. Several of these sites are still in the process of starting up and working with their ethics review boards and at the site level to get ready.
Okay.
But we understand the urgency and we support you in trying to get this as quickly as possible. And so if you just get hold of us, I don't know whose email you gave, Christian, whether it's me or Henk, but maybe to Henk, he's right there in Boston, and he can help get this to the right people immediately.
Yeah, you know what? I can work with Henk and with Seth as well, who couldn't join us tonight in patient advocacy.
Yeah, I think Seth would be an excellent person for this. Yes.
And you can always use the website medinfo@prilenia.com. Then it will always get to the right people.
Yep. Thank you. I just dropped that information straight from your expanded access page.
Yeah.
Thanks, Christian. Christian, do you want me to take the next one?
Please, yeah.
Well, this is a tongue twister, so don't judge. So I'll do my best. D-hydroepiandrosterone sulfate is a sigma-1 receptor agonist. Did you study this molecule?
No. It's the molecule we studied is the most potent selective sigma-1 agonist. Many of these other sigma agonists are out there, but they're very non-selective. In other words, they touch sigma-1, but often have other targets that are totally unrelated. So we're looking, and sometimes the unrelated targets can have side effects. We believe that the selectivity, as well as the potency on sigma-1, is really what gives this molecule its neuroprotective effects and its safety associated with it. Many other drugs do touch sigma-1, but are not really all that potent and actually are often more active on other targets. So no, we did not. This was just sigma-1 itself.
Thank you.
And looking at Pridopodine, that's what it's fairly, I would say, very unique. It is the most potent selective sigma-1 agonist that is out there.
Thank you.
I'll jump into one here. In phase 2 studies, Pridopodine failed to meet primary and secondary bulbar objectives of extending life, secondary objectives of extending life and lowering ALS FRSR in general. Is bulbar benefit the only benefit, or how else are you thinking about the results?
So that in the primary and secondary was in the total population. The mean duration of that population was 22 months with a standard deviation of 8 months. So 60% of patients had 30 months duration. So that's already a significant number in terms of duration. When we looked at those patients who were faster progressors in a short study, which is 24 weeks, we saw improvement in ALS-FRS, in speech, in respiration, and in bulbar function. And so this is all about the population you studied in the very short time of that particular study. If you're studying a group that has a long duration and there's no progression in your 24 weeks, you have no window to see an effect of your drug because they're not showing any progression. And that's why when you look at this group that had faster progression, we saw the effects. The expanded access now that'll go on for another 2 years throughout the United States will give us more impact because it's longer duration, almost 4 times the duration in the larger population, and we'll learn more about the generalizability of our study.
Thank you so much.
Are you noticing, another question, are you noticing any gender differences in the study?
No, we see no gender differences at all. Also, I might say it also has some impact on preserving body weight. Not a massive effect, but we saw some improvement in body weight compared to placebo in this study. Yes. That may be related to swallowing and improvement associated with it.
Yes. Thank you.
Does Pridopodine have any effect on limb onset ALS? And if not, why not?
Well, again, there weren't enough patients who looked at this. We looked at patients with all types of onset, and we saw the effect. We did see in the ALS-FRS, looking at motor function, there was an improvement, but it was less obvious than in respiratory and bulbar function. So there was improvement there, but the biggest effect was in respiratory and bulbar function. And of course, in a bigger study now, we'll be able to understand this much greater in a bigger proportion of patients.
Thank you.
Thank you.
Is there, sorry, one last one. As we think about the fact that that Pridopodine is also being studied in Huntington's, is there any interest as well in Parkinson's or other motor neuron diseases?
Yes, and we do know that the Michael J. Fox Foundation has also highlighted sigma-1 as a target. We have looked in animal studies of Parkinson's disease, both in vitro and in vivo, and we saw benefits, significant benefits in the animal model for Parkinson's disease. We're a tiny company. We're around 30 people, and certainly we don't have the ability at this moment to further extend this to other indications. But it is important because this drug also, by the way, showed protective effects in animals with Alzheimer's disease, animal models, pre-senile in mice. So we see effects much more broadly, but at this stage, we are only able to focus on the rare diseases, Huntington's and ALS, and that will be our immediate focus. But in the future, other indications certainly are worthy of further exploration.
Thank you. Do you anticipate, also, there was a question about timing. Do you anticipate, when do you anticipate phase 3 launching with people going into clinic from a general standpoint?
Henk?
I think we are working to get the trial design agreed and approved by the end of this year, and I would expect first patients being able to access treatment early next year.
All right. Thank you. How do we do, Martha? Do we have any other questions we need to cover?
No, I think that we've had a lot of questions. It's just incredible. We could go all night, but I think that pretty much wraps it up. I think
we just got one more. Do you think it's hypothetically possible that a treatment could be found that would work for those who have been with ALS for more than 5 years and have lost most of their function?
Yes, I think that whatever neurons are still preserved, if we can continue to preserve them, we may not reverse function, but we may be able to maintain stability. So that would be our hope. And that's why early treatment is better, because if you have protection, we're not generating new neurons, but we are protecting them from ongoing injury. And so I believe that keeping people stable would still be very beneficial if we could do that. And it's theoretically possible, and our expanded access will give us more data in that regard.
Thank you. I think we did miss one from somebody from our audience about any kidney issues, discontinuing due to kidney issues. Was this a known adverse event?
No, there was nobody stopping the drug because of any kidney issues at all. No. So there was no serious adverse events with kidney. The drug is excreted by the kidney, and certainly for patients who have renal, and the drug is allowed in mild and moderate renal failure, but in severe renal failure, the drug is contraindicated because you could get much higher levels because the drug's excreted by the kidney. Now, in ALS and Huntington disease, for example, there's no reason, there's no increased incidence of chronic renal disease, but if it's mild or moderate, that would be fine. Severe would be a contraindication because you may end up with much higher levels because it's excreted by the kidney.
Yes.
Thank you very much.
Thank you, Christian. I did see one, if you don't mind, if we have time for one more.
Sure.
Did the Pridopodine, did you notice any effects with limb onset ALS?
Yes, and we've said that we saw in general improvement in all patients. We saw particular effects on respiration and bulbar, even in patients with limb onset, as long as there were some abnormalities in respiratory and bulbar function. And what I said before is our muscle strength and motor, both fine and motor, showed some effect, but were less affected than the benefit seen in respiration and bulbar function.
Very exciting. Thank you.
Great. Thank you. All right. As we mentioned, we work with.
May I just thank all of you? I mean, this is amazing. The level of questions and the number of questions is quite overwhelming. This community is so inspiring, so courageous, and I just want to thank you all for your interest, your encouragement, and our job is to fulfill the hope and really learn more so we can see if this drug offers and can fulfill the promise that we've seen so far.
Thank you, Michael. I think your dedication to ALS and the ALS community is commendable, and having you and Henk and all of you having effort, putting all your effort to bring treatment for ALS is very heartwarming for us. So we can't thank you enough from the bottom of our heart to actually be here and also work tirelessly to bring treatment to the people who are affected. So thank you.
Thank you.
Thank you.
You remain an inspiration for us too, as we work with you and understand your history and your future as you work together, bringing so much intelligence, creativity, and innovation to everything you do. It's very inspiring too.
Thank you. We have a meeting tomorrow, so we have some good news. So
very good.
Well, I'll also thank you for keeping our streak alive of these talks regularly go over the 1 hour expected time. And that's a testament both to an incredibly active community and curious community who are fearless in asking the right questions, and which is any question they have, and to you and our experts who come in and talk and stay for however long you feel you need to, and you always do. So thank you.
Thank you all.
Thank you all. And as a reminder, if anytime you have additional questions, feel free to reach out through Everything ALS or to us. We are always happy to help out.
For sure.
Thank you, Henk.
Good night, and thank you, everybody.
Thank you. Have a great night.
Thank you so much.
Thank you.
And Dee Dee wants to give you a very special thank you with a big smile. That's right, Dee Dee. You made her evening, folks. Thank you, Henk, Michael. Our group is always looking for hope. And when we see people like you so dedicated to your craft, it just inspires us to know somebody's out there thinking about us, and they might even stay up late at night. So, folks, another presentation of Hope Made Manifest. Well, then I'm going to give a couple of last words to Tim. What a pleasure to meet you tonight, Tim. And for Chuck Shaw and his angel to be visiting with us tonight. It always makes my heart warm. Thank you, Chuck. And for all of you who come, it really means a lot to Everything ALS. that you consider us your extended family, because we do this because you're our extended family. And believe it. And with that, pick out somebody who you'd like to say goodnight to tonight. Like, I want to say goodnight to Chuck Shaw. So take yourself off mute if you can. Or if we could.
Good night, Deborah.
Night, Debbie.
Good night, Tony and Sherry.
Good night,
Kristen. Good night, Penny.
Good night, McFinn.
Good night, everyone.
Good night, Martha.
Good night, Janie. Good night, Kelly.
Christian.
Good night, everybody.
Marty. Hi, dear.
Sending you hearts.
Good night, Indu.
Brian Kelly, we'll see you in a second, hopefully.
Good night, Eddie.
Thank you, Eddie, for always being here.
Yep.
Good night,
Jim and Patty.
See you next time.
Brian Wilson, you are awesome. Please come
Good
back
night,
and give
Mona.
us some more of it. And Jubilant, I always say your name every time because I like just saying Jubilant. It's pretty cool. Thank you to your mom and dad.
Good night, Janie. Good to see you.
Good night, everyone. Good
And
night,
Paul
Paul.
Beach, thank you for sharing your lovely partner with us, Paul. Your wife is an angel, and we know it. So, wow.
Good night, McFinn.
All right,
night.
brother.
Good night, Paul.
Thank you so much. Thank you, Kim, David, Carl, Deborah, Zoom user. Cool enough.
Good night, Zoomy.
Love you, Kim, and all the animals that you take care of. All right, everybody. Bye-bye.
Word.